I'm a master's student at National Taiwan University, Department of Horticulture. I'm working at Dr. Alex Cheng's lab in the floriculture section. So Dr. Alex Cheng does research mostly on orchid crops. So the work I'm going to present today is a result of work done by 3 master's students, including myself. So we are investigating the nitrogen-zinc source relationship in Phalaenopsis by using nitrogen-15 as a tracer. Firstly, we would like to thank National Science Council of Taiwan for providing the funding of this research, and also Stable Isotope Facility at UC Davis for performing the nitrogen-15 analysis. Last but not least, we would like to thank our colleagues and friends at Floriculture Hall, National Taiwan University for their material support and emotional support. So Phalaenopsis is our material. It's a very important flower crop nowadays. It has a very long blooming period. It's very easy to control the flowering, and also it's very well adapted to indoor use. That's why it's now very popular. As some of you may know, orchids are very resilient crops. They are very resistant to drought and also to nutrient stresses. And in some orchids, the pseudobulbs, which are enlarged stems, they can store water and nutrients. That's why they are so resilient. Phalaenopsis doesn't have pseudobulbs. However, we also find that it's very resilient as well. Previous workers, for example, Lei, a previous worker at Dr. Alex Cheng's lab, also found that Phalaenopsis is very slow to show deficiency symptoms. One of my classmates, Yulia Chi, is also presenting a poster in this conference, number 262, and her results also found similar trends. And so Phalaenopsis is very slow to develop deficiency symptoms, including nitrogen. And so this suggests that there is a large pool of stored nitrogen in Phalaenopsis. The question is, where is this nitrogen stored? Well, we think it's probably in the leaves and the roots, because in some Phalaenopsis species, they go dormant in winter. And during this period of dormancy, they lose the leaves, and for the subsequent growth, they use storage from the roots. In many Phalaenopsis species, the leaves are very thick and fleshy, and so probably the leaves have similar function. And so we observed these observations, but currently there are no published data to support these. So the research objectives, the current observations show that Phalaenopsis have significant pool of stored nitrogen. But so far, there are no experimental data to support these. So there are other questions we want to get answered. What are the major storage organs in Phalaenopsis for nitrogen? What are the sinks? And lastly, what is the sink-salt relationship among the various organs? So to answer these questions, we used nitrogen-15 to trace the movement of nitrogen. Nitrogen-15 is a stable isotope, and so it doesn't, it's not radioactive, so to trace it, we don't use radiography. Instead, we use mass spectrometry. So to do nitrogen-15 labeling, there are a few methods. The method we are using is the enrichment method. In this method, we add a nitrogen-15 enriched material into a system with natural abundance. And so the system will get enriched with nitrogen-15. And so with Phalaenopsis, the method is we added nitrogen-15 through the fertilizer. So we label the fertilizer. After a period of cultivation, the nitrogen-15 label will be distributed among the various organs, and we can analyze them separately to trace the movement of the fertilizer. And so in a nutshell, the findings of this study is as follows. We show the significance of stored nitrogen pool in Phalaenopsis. Phalaenopsis leaves and roots can store nitrogen for later use. Newly grown leaves, newly grown roots, and the developing inflorescence are strong sinks for nitrogen. So we are going to show you the results in a minute. In the first experiment, we investigated the fate of recently absorbed nitrogen in the leaves, in the actively growing leaves. And so we used freshly repotted plants. We added fertilizer, we added labeled fertilizer, and then after a period of cultivation of several days, we harvested the first leaf and the fourth leaves. The purpose of harvesting the first leaf and fourth leaf is to compare the amount of fertilizer nitrogen accumulated, and also the purpose is to analyze the contribution of fertilizer nitrogen for the growth of the young leaves. And so the result is we found that the first leaf is actively growing during this period, and the first leaf accumulated dry weight significantly. And also the nitrogen content also increased significantly during the 8-day period. So the increase in nitrogen content was 4.3 milligrams. And this graph shows the increase in nitrogen-15 content excess. Well, the label can represent the fertilizer nitrogen because we add the label to the fertilizer. And so from this result, we found that in the first leaves, fertilizer nitrogen was accumulated significantly. And the increase during the 8-day period was 100.70.9 micrograms. And so we calculated the ratio of the label to the fertilizer, and we found that the fertilizer nitrogen accumulated was 4.5 milligrams. And this is a very small number, only 12% compared to the 4.3 milligrams accumulated in the first leaf. 4.3 is the total nitrogen, and 4.5 is the fertilizer nitrogen. And so from this experiment, we found that only 12% of the nitrogen accumulated in young leaves came from the fertilizer. And so even though fertilizer nitrogen was available and absorbed from the roots, to meet the requirements of young leaves, nitrogen still had to be sourced from elsewhere within the plant. And so we found that stored nitrogen pool plays a very significant role to supply requirements of growing tissue in Phalaenopsis. In the following experiment, we investigated the effect of leaf age on the zinc strength for nitrogen. And so the plant used is like this pictured here. We labeled the leaves from 1 to 6, 1 being the youngest leaf. And so we added labeled fertilizer to the roots. And after 8 weeks, all the leaves were harvested and analyzed for nitrogen-15. So from this table shows the partitioning of fertilizer nitrogen in leaves after 8 weeks. The columns on the left shows total nitrogen concentration and content, but what we are interested in is the fertilizer nitrogen. So we see the nitrogen-15 concentration and content. From these two columns, we found that the first leaf contained the highest nitrogen-15 concentration and content. And so these two parameters decreased as leaf age increased. And so the conclusion from this experiment is that zinc strength for nitrogen decreased as leaf age increased. In the following experiment, we investigated the translocation of fertilizer nitrogen after foliar or root application. And so the plant's views were unpotted, and the roots were separated into old roots and new roots. And labeled fertilizer was applied at different organs, firstly on the upper and lower leaf surfaces, and also on the old roots and new roots. And so the plants were grown on. And after a period of 8 weeks, we investigated the distribution of the translocated nitrogen. The values in blue are high because that's the value of nitrogen-15 where we applied the fertilizer nitrogen. So for example, we applied fertilizer nitrogen in the mature leaves, so it's high in the mature leaves. But the unshaded values are where the amount of translocated nitrogen in the various organs. So after applying at mature leaves, they were translocated to newly grown leaves, roots, old roots, or newly grown roots. So we found a very interesting result is that regardless of where we add the labeled fertilizer, the nitrogen-15 concentration was consistently highest in the newly grown leaves and newly grown roots. So what does this tell us? It tells us that newly grown leaves and roots were stronger sinks for nitrogen compared with all the other organs. In the next experiment, we conducted root excision. What is the purpose of removing the roots? Well, this is to, firstly, we grow the plant, and then we added labeled fertilizer, so they are stored. We give the plant time to store the fertilizer nitrogen in the various organs. And we remove some of the roots to remove reduce the size of the nitrogen pool. And so we remove from 0 to 75% of the roots to reduce the nitrogen pool. And then after that, we induce the plant for flowering, and then to investigate the distribution of nitrogen. So root excision corresponds to reduction in nitrogen pool. So we found a very interesting result is that regardless of reduction in nitrogen pool, newly grown leaves and inflorescence got the similar allocations of the stored nitrogen. Here, nitrogen 15 represents stored nitrogen. And so because the values are similar, we found that they are strong sinks of nitrogen. But in the mature leaves, the amount of stored nitrogen decreased as more roots were removed. So this shows that mature leaves can store nitrogen because they release some of them afterwards. So the findings of the result is that even though stored nitrogen pool was reduced, newly grown leaves and flower stalks received similar allocation of stored nitrogen. So the newly grown leaves and flower stalks are strong nitrogen sinks. And also, more importantly, in this experiment, we demonstrate that mature leaves can store nitrogen and later function as nitrogen source. In the last experiment, we investigated the absorption and partitioning of nitrogen over vegetative to reproductive period. So young plants were used, and then similarly, labeled fertilizer was applied to the roots. And the plants were grown for 38 weeks until they were flowering at the until half of the flowers were open. And so they were sampled from time to time, from the first week to the 38th week. And so the result is that from this graph, we can see that the roots can store nitrogen. How do we see that? So nitrogen was accumulated in the roots from between week 0 to week 8. But afterwards, the concentration of nitrogen 15 decreased, especially after flowering. And so what does this tell us? It tells us that nitrogen was stored in the roots and then later shipped elsewhere for use in other organs. Similarly, in the leaves, we also see a similar pattern. So it increased and then decreased. Also, from this graph, we can see that newly grown leaves and the inflorescence both increase in nitrogen-15 content despite that the other organs decrease in nitrogen-15 content. So they are strong sinks of nitrogen. So the findings of this last experiment is that nitrogen-15 concentration of roots decreased from week 8 onwards, while the value of other organs still increased. So this shows that the roots can function as nitrogen source. Similarly, we found that the leaves can also function as nitrogen source. Lastly, we found that the new leaf and inflorescence, they were consistently increasing in nitrogen content, and so they are strong sinks for nitrogen. So to conclude, we have demonstrated the significance of stored nitrogen pool in Phalaenopsis. We have also shown that mature leaves and roots can store nitrogen, which can be translocated when needed at a later time, thus showing the function of these organs as nitrogen source. So the newly grown leaves, newly grown roots, and developing inflorescence are the strong sinks for nitrogen compared with the other organs. So that's my presentation. I've got a couple of questions. Thank you.
Question? Yes. Just a question. So when the flowers themselves senesce, does nitrogen get removed once back out of the flowers or of the petals? Have you looked at that?
Well, we did not study that yet, but in some other crops it has been shown. So probably that will be our study.
Are you imposing deficiency, a nitrogen deficiency after this? I guess where I'm confused is that we know nitrogen is mobile, and you're showing that it's moving around the plant, which inherently isn't really that surprising that it's moving to where it's needed. And so I'm wondering, are you restricting nitrogen so that you see that it's moving, or is there a steady supply of nitrogen throughout the experiment?
In the above experiment, they were supplied with nitrogen all the time. But we conducted separate experiments where we limited the amount of nitrogen. And yes, it's separate sets of experiments. But we see that it's very interesting in Phalaenopsis that when we limit nitrogen, it takes them a very long time to show symptoms. So in other studies, we studied how nitrogen is translocated when they are limited of nitrogen despite the absence of symptoms, but not in this presentation.
Thank you very much. Well, that concludes our session. I do believe the speakers, I think everyone is still here. So if you want to ask questions of the speakers after we're done here. But thank you very much for your attention today, and I hope the rest of your stay here in Miami is bright. Thank you.